We are now live on air for Charles Pepe-Ranney's talk. Chuck did his bachelor's and PhD at the Colorado School of Mines with John Spear, and so we know his talk will not suck. If you don't know John Spear, that is his catchphrase. This does not suck. Chuck is now up at Cornell doing a postdoc with Dan Buckley, who I meant to tell you I still want to apologize. I hit him really hard in the head with a volleyball like 10 years ago, and I still feel
Oh, yeah?
But he's going to do a really cool talk today on stabilized isotope probing in soils, and we warned him that he might have to do a second talk on his methodology for giving his presentation because he's going to show you some really cool stuff, so hopefully he'll tell us a little bit about that. So with that, Chuck, if you want to take it away and share your screen, we'll get going on the talk. A reminder to everyone that you can ask questions inside the Hangout or ask them on Twitter using the hashtag #USeminar for microseminar.
Okay. All right, can everybody see that?
Let's get it.
Okay, cool. So first, thanks to Jen and the other organizers for inviting me. This is really fun. I think the micro seminar is great, and it's really awesome that Jen and Cameron and David are putting it together. So this is fun to be here. Yeah, I'm a postdoc in Dan Buckley's group, and in Dan's lab, we're interested in defining the organization of the soil microbial ecosystem. And we're sort of interested in that because soil is this large reservoir of carbon, and soil microorganisms are driving the flux of carbon through the soil carbon reservoir, and therefore they have a large effect on, say, the flux of carbon from plant vegetation into soil and then to the atmosphere. And if we want to understand how, say, climate change or land management will impact soil organic matter, then we need to understand soil microbial ecology. But it's quite difficult to study soil microorganisms, frustratingly. And I say frustratingly because soil is just so accessible, and it's intuitively important. And I think this figure from this paper by Pat Schloss that's up on bioRxiv really highlights some of the difficulties of studying soil. So here we have a rarefaction curve of small subunit ribosomal RNA gene sequences from databases, categorized by different environments. And soil is this blue line on the top here. And what you should note is that the soil line is steeper than the others, which indicates that soil has relatively more diversity than the other environments shown here, and that presents problems for soil microbial ecologists. The other thing that you should note is that the soil line is shorter than some of these other environments, and that tells us that there are less reference sequences in soil than there are for, say, the aquatic environment, which also presents challenges. And it's especially difficult because soil is so diverse. And they highlight this in the paper, saying that the orders of the categories is surprising, considering how much biodiversity is harbored. I should say the Earth's biodiversity is harbored by the soil environment. Right. So what that means is that we don't know much about the implications of some of the ecological phenomena that we observe as soil microbial ecologists. I should say specifically, we don't know much about the biogeochemical implications of these phenomena. And I think this paper by Noah Fewer's group really highlights that problem. So this is a figure from a paper out of Fewer's lab where they surveyed verruca microbial diversity across a variety of soil biomes. And they found that verruca microbia are essentially everywhere and often in really high abundance in soil. And so on the x-axis here we have different biomes, and on the y-axis we have relative abundance or just relative abundance. And so the height of the bars denotes the relative abundance of Verruca microbia in the biome. And this last bar here on the right is the only sort of managed ecosystem. So these are sort of more natural ecosystems, whereas this bar on the right is agricultural soil. And there's this association, or I should say there's this effect of land management on soil microbial or soil Verruca microbial abundance. And this has been noted by other groups as well. But we don't know much about the implications of this phenomenon because we don't know much about the functions of soil Verruca microbia. And we don't have many of these soil Verruca microbial phylotypes in culture. In fact, they're very distantly related to things that we have in culture. And this is highlighted really well in this paper. Okay, so soil is really diverse. We don't have a lot of references to call on. And so if we're going to study soil microbial diversity, we need a method that scales to the diversity and allows us to observe activity. And in Dan's lab, we think that one of these methods is, or a good method for this purpose is DNA stable isotope probing, or SIP. And SIP is a method that allows you to sort of connect genetic identity to function in natural microbial communities without having to culture the microorganisms. So I'm going to sort of briefly run through the SIP methods, and then I'm going to get into one of our studies. So this is a series of images from one of the first really stable isotope probing paper. And the premise of SIP is that if you feed a microorganism a 13C carbon source, it will incorporate that 13C into DNA, and that 13C DNA can be distinguished from 12C DNA on the basis of buoyant density. And so this left image here is really a proof of concept for that premise. So each of these tubes has about, I don't know, 25 mils or so of cesium chloride, and the tubes have been spun such that we've created, or they've created a density of cesium chloride within the tube with low densities on top and high densities on the bottom. Okay, and the bands here are the positions of DNA. And this left tube, in this left tube, 12C DNA from a methylotrope culture has been added, and you can see that it falls about at this position on the buoyant density gradient, or excuse me, on the density gradient. And in the right tube is the same organism that's been fed 13C methanol, so now it has 13C DNA, and its DNA falls out down here at higher buoyant density. Right, so that is great. But when we're studying natural communities, we're really trying to target a subset of the whole. And that's what this image, this second image, is depicting. So the DNA in these tubes are from soil. And you can see right off the bat that instead of these nice clean bands, you have this sort of smear of DNA. And this smearing is due to GC and diffusion. Okay, and there's a couple things I want to point out. Right? You can have microorganisms in high abundance in the high GC region of the gradient if the organisms have, excuse me, in the high density regions of the gradient if the organisms have high GC, because GC content is positively correlated with buoyant density of DNA, right? So you can also have organisms in high abundance out here, or their DNA in high abundance out here, if they're in high abundance in the community, even when they're low GC because of diffusion. Okay, so when you're trying to define labeled microorganisms from unlabeled microorganisms in a DNA SIP study, it's really important to compare high density fractions from your 13C gradient to your 12C gradient. And only in that comparison can you unambiguously detect labeled microbes. So the game is really figuring out what's been enriched out here in your 13C, or the gradient of DNA from your 13C treatment versus your 12C treatment. So yeah, you're comparing high-density fractions between control and labeled treatment. And this is sort of the principle behind our experimental design for the project that I'm going to present in a little bit. Okay, so this is just like an image of some real data for a single OTU. And we're showing this OTU assayed at multiple time points and its relative abundance across these cesium chloride density gradients based on its representation in ribosomal RNA gene libraries. So the color of the line tells you the day at which we did the survey, and then the shading under the line tells you whether the relative abundance value is from DNA from a 13C treatment or from a control treatment. So orange is control and green is soil where we've added, this is relative abundance for an OTU in soil where we've added 13C xylose. And this is a microbe that we believe was labeled in our experiment, and you can see that its DNA out here, or the DNA out here was enriched for this microbe at days 1, 3, and maybe 7, but not so much at days 14 and 30, because at days 1 and 3 in particular, it has higher relative abundance than the control. Okay? And there's lots of papers out there on DNASIP methods. I know Josh Neufeld is in the Hangout, and he's pioneered a lot of these methods. So I would certainly read papers by Neufeld. I think Jay Lennon has some good DNASIP papers out there, in addition to Ralph Conrad's group. I know I'm going to miss some groups, and Bruce Hungate has also done a really good job of pioneering some DNASIP methods. So those are the groups that I would first look at if you're trying to understand how the process works. And this is a nice paper that came out very recently, Environmental Microbiology Reports, which sort of highlights how high throughput sequencing enhances stable isotope probing. Okay, and sort of like the holy grail for the experiments that we're doing would be to define the activity of globally cosmopolitan soil bacterial lineages, like the DA1 Verruca microbiome that Noah Fear's group has been studying. And I just saw this paper came out recently about the DA101 genome. So DA101 is this Verruca microbiome that we've known for a long time is ubiquitous in soil and often in really high abundance, but it's not been known what sort of ecosystem functions it performs. So it's great to have this genome, and it would be nice to define its activity in SIP experiments, for instance, or the activity of other microorganisms that are ubiquitous like DA101. Okay, so the other thing that we'd like to do is to sort of build a conceptual model for the food web of different soil ecosystems. So this is just an old sort of detrital food web from an entomology paper that I found in 1988, and you can see that the bacteria represent this kind of single node here. But as soil microbial ecologists, we see this node more like this, with just like an amazing wealth of diversity just in the soil bacteria alone, and much of the carbon throughput in soil is being carried out by these different soil microorganisms, and we'd like to figure out how they're organized in relation to soil carbon cycling. So this is just kind of a conceptual model from the experiment I'm going to show you, and we'd like to kind of do this for lots of different soils and see if there are common themes in ecology from soil to soil or not. And we'd also like to see if there are microorganisms that are performing similar functions, or different types of microorganisms that are performing the same function in different soils, and if those microorganisms are also found, to be globally distributed. Okay, so I would have had a much harder time in my postdoc if it wasn't for our grad student Ashley Campbell. She's awesome, and she's I should say she's our former grad student. She's now a postdoc at Lawrence Livermore National Laboratory, and the study I'm going to show you is a collaboration between me and Ashley. So thanks, Ashley. And the experiment that Ashley and I worked on was one where we tracked 13C from xylose or cellulose into a soil microbial community over time. And cellulose is obviously a really abundant component of plant biomass, and so is xylose, and so that's why we chose these two substrates. But you could really do this experiment with a number of different carbon sources. And this is a more detailed view of our experiment. So we had a control treatment where we added all 12C, or only, I should say, 12C substrates, and we had a label treatment where we singled out xylose to carry our isotopic label, and we also had a label treatment where we singled out cellulose to carry the isotopic label. What's important to note here is that for every treatment and every time point that we collected a sample, we have 20 density gradient fractions. We have, I should say, we have small subunit ribosomal RNA gene libraries from 20 density gradient fractions. So we're profiling the entire gradient for every sample, every treatment, every time point. Okay, so this figure here is an ordination of all of those small subunit ribosomal RNA gene libraries. Okay, so every point here represents a density gradient fraction. Okay, so this density gradient fraction here that I'm hovering over was from our day 30 collection at a density of 1.732. And the blue points on the left panel here are from gradients from DNA that was extracted from treatments that received the 13C cellulose amendment. So there's a couple things I want to point out. First, orange on this left panel denotes the control gradient fractions. And you can see that both in the control gradient fractions and in the gradient fractions from treatments that receive 13C cellulose and 13C xylose, which is in green here, density plays a pretty major role in structuring the profile of ribosomal RNA gene sequences in a fraction, and that's because of GC. So out here, low GC organisms are enriched, or the DNA in the gradient fractions out here is enriched for low GC microorganisms, and out here the DNA is enriched for high GC microorganisms. But we saw in the heavy fractions our samples, our gradient fractions diverge from control for both the cellulose treatment and the xylose treatment. And we can tell, based on this ordination, that the kinds of microbes that are labeled in the cellulose treatment are different from the kinds of microbes that are labeled in the xylose treatment because their compositions are different. And if we look at, say, the cellulose gradient fractions from the high density region, we can see that they diverge from control at day 30. So on the right panel, I've colored the points by the day of that the sample was taken. So here is a gradient fraction from day 30, and here is a gradient fraction from day 14. So the cellulose diverged from control at days 14 and 30, whereas the xylose treatment DNA diverged from control at days 1, 3, and 7. And if we zoom in on the xylose, we can see that there's also this like temporal variation in the activity. So the types of microorganisms that were labeled at day 1 are different from those that were labeled at day 3, and which are different from those that were labeled at day 7. So we're seeing this kind of cascade in activity that's apparent when you ordinate all of the small cell-unit ribosomal RNA gene profiles. Okay, so let's dig into which kinds of microorganisms are driving these differences in the profiles, or the density gradient profiles. Okay, this is showing an enrichment value for all the OTUs that we found in our experiment. So each point represents an OTU. OTUs that are high up here on the y-axis, these guys, are the ones that are labeled. I should turn the hover off here. Right, so these are the ones that are labeled in the cellulose treatment, and then these are the OTUs that are labeled in the xylose treatment. Okay, I filtered this figure to only show the maximum enrichment value at any time point for an OTU, and then we have the cellulose up top and the xylose on the bottom. So just to be clear, if an OTU is up here, it's enriched in the high density fractions from the treatment, the 13C treatment density gradient relative to control. And you can tell because I'm only showing the maximum enrichment over time for any given OTU that excuse me, I should say like the OTUs are colored by the time at which they had this maximum enrichment value. So the blue and green colors in this cellulose panel denote days 14 and 30, and the colors in the panels on the xylose denote days 1, 3, and 7. And that's telling us just like the same thing that we saw in the ordination, that the cellulose organisms were the organisms that were labeled by 13C cellulose were labeled at days 14 and 30, whereas the organisms that were labeled in response to 13C xylose additions were labeled at days 1, 3, and 7. And a couple things I want to point out that that one of the interesting things about the xylose treatment was that many of these OTUs were from lineages that are abundant and fairly ubiquitous in soil, like verruca microbial abundances. We know from that study I presented earlier that out of Noah Fear's group that verruca microbia are abundant in soil, and we saw many verruca microbia that were labeled in response to 13C cellulose additions. And the xylose, like I pointed out in our ordination figure, responders, or the microorganisms that were labeled in response to 13C xylose, changed over time, right? So we saw at day 7 these actinobacteria types that were labeled, but at day 3 we saw these Bacteroidetes types labeled, and then at day 1 it was these Firmicutes types. And I'm going to go into that a little bit further, but I also want to point out that these are 3 OTUs that I'm highlighting. This is OTU 120, which is a member of essentially a non-phototrophic sister clade to cyanobacteria. This is OTU 6, and that is a Cellvibrio OTU, and Cellvibrio appeared to be labeled in our experiment. And it's a canonical cellulose degrader, so that was a nice sanity check. And then we saw Planktomycetes OTUs. This is OTU 50 or 150, that were labeled on experiment as well. But before I get into these specific OTUs, I want to talk a little bit more about this, the temporal activity, or the dynamic temporal activity that we observed in the Xylus treatment. All right, so now I'm just taking those same fold change values that we saw above, and just looking at the data for these three taxa: Firmicutes, Bacteroidetes, and Actinobacteria. And I'm showing, instead of just the maximum fold change for each OTU, I'm showing fold change for days 1, 3, and 7. Okay, so at day 1, you can see these Firmicutes types are labeled here. And these are all Penibacillus, which is a spore former. It's known to grow fast. It has lots of ribosomal RNA operons. And at day 3, we saw Bacteroidetes types labeled. But we didn't see our Firmicutes types labeled anymore, right? So it seems as if the label was exchanged between the Firmicutes to the Bacteroidetes here. Okay, and then at day 7, we saw Actinobacteria labeled, but our Bacteroidetes were no longer labeled, and neither were our Firmicutes, right? So this, to us, suggested a trophic exchange of carbon from, say, Firmicutes to Bacteroidetes, and then maybe from Bacteroidetes to Actinobacteria. Further, not confirming that hypothesis, but further evidence for trophic exchange came from the phylogenetic affiliation of these OTUs. So the Actinobacteria that we found labeled at day 7 were closely related to a microbe called Agromyces ramosis, which is a known predator in soil, and it's actually fairly ubiquitous in cosmopolitan, and it's been studied, and it's been grown in culture, although I think it's fairly fastidious. So you know, we have this hypothesis that you know, the degradation of xylose in our experiment was characterized by trophic carbon exchange. And it's really important to define these interactions between microbes and the soil microbe community, because these interactions can mediate some of these climate change feedbacks that we might expect. So if climate change alleviates bottom-up control on microbial growth, then predators like maybe Agromyces ramosis or nematodes, for instance, might mediate that. And so this is just a nice paper that where a group removed predatory animals from soil. I mean, obviously we won't be able to do the similar experiment with Agromyces ramosis, and then alleviated these bottom-up controls on fungal growth, and showed that when you remove the predators, the fungal growth just continues to increase as you add more resources. But if you keep the predators in the soil, the fungal abundance stays pretty constant. So this is out of Mark Bradford's lab and was published in PNAS in 2015. You know, it would be nice to really understand these interspecific interactions in soil, so we can get a handle on how these, or what interactions might be able to mediate feedbacks to climate change. Okay, so I also want to point out that Thea Whitman, who's now at University of Wisconsin-Madison, did a study out here in upstate New York in collaboration with our lab, where she added fresh corn stover to soil and looked at how that altered the microbial community relative to just a control soil where she didn't add any biomass. And she sort of tracked what happened to the microbial community over time. And at day 11, she saw this bloom of Firmicutes. Excuse me, I think it was day 12, but it rained on day 11. So these microorganisms that bloomed in the Stover fresh corn biomass amendments were Penibacillus, just like we saw responding to the xylose that we added. So we're seeing phenomena that are consistent with what we saw in our. We're seeing environmental phenomena that are consistent with what we've observed in our SIP microcosms. Okay, so ultimately, I've shown this figure before. You know, we'd like to get to where we can start to build conceptual models of food webs for different soil microbial communities. And so, you know, like to summarize, we saw xylose enter the soil microbial community via the activity of these Penibacillus types in the Firmicutes. And then we saw Bacteroidetes become labeled, and then we saw actinobacteria become labeled. But we can't say for sure whether these Bacteroidetes were labeled in response to, you know, primary degradation of xylose or predatory interactions or just feeding on labeled Penibacillus biomass. And neither can we say for sure that actinobacteria became labeled due to predatory interactions. But given the phylogenetic affiliation of these types, I should point out that I think there's 4 bacterial phyla that harbor predators, and 2 of them are the Bacteroidetes and the actinobacteria. But you know, given the phylogenetic affiliation of the microbes that we saw labeled in our experiment and the cascade in activity, that's an interesting hypothesis to pursue. And then we saw the cellulose being degraded by cosmopolitan lineages within soil, including Spartobacteria and the Roucomicrobia and different kinds of Planktomycetes and Chloroflexi. And then we took each OTU, and I'm just going to blow through this slide. So we took each OTU that we found to be labeled in response to either the addition of 13C cellulose, which is the blue in all these figures, or xylose, which is the green. And we kind of described those OTUs both based on their abundance in the bulk soil in our microcosms, based on predicted ribosomal RNA gene operon number, and we tried to assess how much these microorganisms might have shifted in response to 13C labeling. I should say how much their buoyant density might have shifted in response to 13C labeling. So if a microbe's DNA is sort of heavily labeled and it shifts a lot, then that might be indicative of a higher substrate specificity than a microbe that's DNA wasn't so heavily labeled. It could be, say, consuming both your labeled substrate that you added in addition to other substrates that were in the amendment or maybe that were available in the soil. So if you want to get more info on some of the specifics of this, you can check out. We have a preprint up on bioRxiv from this study, but to summarize, we found that the microorganisms that were labeled in our xylose treatment had more predicted ribosomal RNA gene operon numbers. It seems as if the cellulose degraders that we identified had higher substrate specificity than the microorganisms that consume xylose. And in general, the microorganisms that consumed xylose were often more abundant in the bulk soil than the microorganisms that consume cellulose. So these are just rank abundance plots of our bulk soil, and the tick marks represent the positions in the rank abundance plot of our responders. Okay, so that's the data from the amplicon sequencing that we did of the SIP gradient fractions. So in addition to doing amplicon sequencing of small subunit ribosomal RNA genes, we took the day 30 high density fractions from the DNA from the soil that received the 13C xylose treatment and did some shotgun metagenomic sequencing. Because we thought, well, if we're enriching this DNA for these microorganisms, maybe we can use that DNA as template for some community genomics, or maybe we can build genomes from the DNA sequences in that pool of DNA. Okay, so if you remember, I did highlight in our fold change plots 3 OTUs that we thought were labeled in this cellulose treatment, and one was OTU6, which was a cell vibrio. One was OTU20, which was a cyanobacterium, or a member of a citriclade cyanobacteria. And one was a Planktomycetes OTU. And I'm just highlighting those 3 here so to have some thread from that data to this. This red line is showing the abundance of the xylose, or excuse me, the cellulose labeled OTUs in the high density fractions versus the abundance of the same OTUs in green here in the bulk DNA. So this is essentially showing the enrichment not between control and label, but the enrichment between bulk DNA and the high density CYP gradient fraction DNA, for microorganisms that we thought were labeled in response to cellulose additions. And so you can see that often we got at least 10-fold enrichment from the bulk DNA of these microorganisms. And so that might help us to essentially get more bang for our buck when we're doing shotgun metagenomic sequencing for these microbes, relative to if we just did sequencing on the bulk soil. Okay, so we took that DNA and we did, the sort of classic community genomics approach, where we assembled the shotgun DNA fragments into contigs, and then we grouped those contigs into genome bins on the basis of oligonucleotide signatures. And this is a figure that's sort of showing how the contigs are organized when you relate them to one another based on pentanucleotide frequency. Okay, so each point in this figure represents a contig from our assembly, and they're colored based on some taxonomic affiliation. So these contigs here sort of have similar pentanucleotide frequency, and they're all from alpha proteobacteria. If we look at this cloud of contigs here, we see that these are all our cell vibrio types in the gamma proteobacteria. So basically, these clouds are nearly complete, sometimes, genomes from DNA that was sequenced from high-density DNASIP gradient fractions. And we found that we were producing genome bins, or bins of really closely related strains from many of the microbes that we found labeled, including this cell vibrio, some of these Planktomycetes types. So this one is that OTU 150 that we talked about earlier. Oh no, this is the cyanobacterium, excuse me. We also found that we have genomes from Verruca microbia types, excuse me, some that appeared labeled in our experiment, and some that did not. And we're still kind of like crunching all this data. We also found some interesting bins of contigs from these really poorly characterized groups, including the Parq bacteria. And then we took all these contigs and obviously, I think, characterized the glycoside hydrolase genes on them, and looked for glycoside hydrolases that are associated with cellulase activity. And so that's what I have in this tab here. Gray contigs do not carry a glycoside hydrolase that's associated with this cellulase activity, and the red contigs are the ones that do carry glycoside hydrolase genes that are associated with cellulase activity. So we see, a lot of cellulases or putative cellulases in the cell vibrio genome, like you would imagine. We see cellulases in our verruco microbial genomes. We see them in this cyanobacterium, this OTU 120 that we highlighted earlier, and a number of the microbes, as you might expect, from the metagenomes that we produce in this study. This is just a bar chart of the substrates that are associated with all the glycoside hydrolase families that we found. We have 3,100 cellulase associated glycoside hydrolase annotations. We produced many genomes that were nearly complete and discrete, without much evidence for contamination. But a lot of the genomes that we produced that were nearly complete and discrete didn't appear to be labeled in our experiment, and we're trying to wrap our heads around why that might be, and it's either a sensitivity issue or a methods problem with our the way we carried out the stable isotope probing, or it's just a reflection of the heterogeneity or the diversity in DNASIP gradient fractions. So I think that often we like to think that when we're doing DNA stable isotope probing that in the high density region we're only seeing DNA from labeled microbes, but in reality, because of diffusion and because of this GC variance of the community, you have lots of DNA from unlabeled microbes out there, and I think this really highlights the importance of doing rigorous controls when you're working with DNASIP. The other thing that I think is cool from this metagenomics study is that we are finding genomes from microbes that are really distantly related from anything that's been well characterized. Okay, so we're finding nearly complete genomes from Park-U bacteria types, soil Park-U bacteria, that are not at all related to anything that we have in culture. Some that share less than 80% sequence identity in a small 7-unit ribosomal RNA gene with anything that we have in culture. We're finding genomes from microbes that are members of cosmopolitan lineages like Verruco microbia and Chloroflexi and Plectomycetes. And we're also finding genomes from microbes like that cell vibrio that are canonical cellulose degraders. So it's a really interesting data set to work with. And the end goal again is this conceptual model. And if we have more evidence, some genomic information from these OTUs that were labeled in response to these substrates that we targeted, then maybe we can start to disentangle how the carbon moved over time from one group to another. And maybe we can confirm the cellulose activity of some of these other types with that genomic information and start to fill out this food web. Okay, so with that, I'd like to do some acknowledgments here. It's been like a real pleasure for me to work in the Buckley lab. We have some awesome grad students. Ashley, Chantal, and Mallory are just amazing. Nick and Spencer are really productive postdocs that work in Dan's group. Nick has done some really cool experiments with lots of different substrates in soil, and Spencer's just getting started, and he's already got a lot of cool data. Thea has been a really great collaborator and friend as well, and this work was funded by the Genome Science Program at the DOE. Okay, that's it. Do we have any questions? I'm going to turn my screen sharing off. Oh. Am I still sharing the screen?
No.
Or...
Okay. Sure. Stop presenting. Okay. Why am I feeding? I'm feeding back to myself.
What's that?
Are you hearing feedback? Oh wait,
no.
You cleared up. Okay. So awesome talk, right? A bunch of questions already came in, and I'll ask if anyone inside the Hangout wants to ask a question, please unmute yourself and pop back up. I should hopefully have released all control to that. So a couple of questions came in over Twitter, so I can ask them initially, if I can find them, right? So Paul Carini was pretty excited that you're already citing the papers from Noah's lab, and so he was asking about, it came before you showed the slide, but he asked about cellulose assimilating verrucos, about their abundance, and were any of them relatively abundant in the experiment? Were cellulose assimilating verrucos relatively abundant, and then were they relatively abundant in the total experiment? You sort of answered that a little, but can you just sort of...
Yeah, I mean, I guess it depends on what you mean by relatively abundant. They weren't all that abundant in our soil, so we're working with an agricultural soil, and that tends to decrease the verrucomicrobial abundance, and they certainly weren't amongst the most abundant cellulose degraders. The most abundant cellulose degrader that we found was this Vibrio OTU, and it was by and large the most abundant microorganism that was labeled in response to cellulose. So yeah, I guess the short answer is no.
Okay. Another question was, so if you want to just tell us a little bit about the Project Jupiter that you're doing. There's a question about is there using the bokeh package?
Yeah, I'm using this really, there's a Python module called bokeh that's developed by the people at Continuum Analytics. It's open source, it's freely available. It's fairly low level. I think it's kind of like ggplot2, although maybe a little bit lower level in terms of the syntax. And I'm using that to make the interactive figures, and they make it so nice to just add things like hover interactivity and zoom, and yeah, so check out bokeh. It's pretty cool. It's pretty slick, and it's really into a great tool.
Cool. Okay, does anyone inside the chat want to unmute themselves and ask any questions? I may have accidentally muted you all forcefully, so just enter in the chat window if you can, if you can't speak up right now. Okay, so apparently John is saying that Titus Brown's work is done if you've done this presentation. Okay, we have a question that came in from Colorado School of Mines. It looks like John Spear's entire lab has an account. So, do you think primary cellulose degraders are also taking up the labeled C, or is there more scavenging by non-cellulose degraders?
So that's an interesting question. That's one of the limitations of CIP is it can be difficult to distinguish between cross-feeders, say, from primary degraders. And I would imagine the cellulose degraders are part of a biofilm that's growing on this insoluble cellulose substrate. So I think that probably quite a few of the microorganisms that we're seeing labeled in our experiment are not acting on the cellulose directly, but I think that it's going to be difficult to distinguish that from the primary cellulose degraders.
Sort of a follow-up question for that. So when you put in your labeled cellulose, do you know what the molecular weight was or what the structure was, and that how many units were repeating in every molecule, or?
Well, I don't know if we know to that detail. It's bacterial cellulose, so it's certainly not in terms of the crystallinity as representative as we would like of plant biomass cellulose, and that certainly might be an artifact that's part of our experimental design. But there's it would be difficult to get cellulose, plant biomass cellulose of the purity. I think maybe Josh has other thoughts on this that to use in these experiments.
Well, so the reason I ask, right, is that a lot of cellulases are extracellular.
Uh-huh.
And so you're going to have primary breakdown. And so it kind of depends. I wonder, actually, in your data, not like you should do this because you got a job and you need to leave, but someone in Dan's lab, right, could look at this. I seem to remember that you can actually tell if it's an endo or an exocellulase, or if there would be a difference.
Like based on the actual glycoside hydrolase.
Yeah, and that, right, because it goes down to cellobiose, right, and then you could look at how many organisms could do cellobiose transport based on your metagenomes.
Yeah, absolutely, and that would be the thing to look for. I think is like if you're a cellulose degrader, you have a cellulase and like a beta glucosidase. But if you're a cheater, then you're going to have a beta glucosidase and probably not a cellulase.
Mm-hmm.
And yeah, and that's something that potentially the metagenomes can help us with. And yeah, I think that's certainly a path that we need to go down. To start looking at other glycoside hydrolase genes that might be indicative of, say, cheating.
Not cheating. It's efficient usage.
Yeah,
right? Yeah.
Yeah.
So with that, another question just came in about Pulsechase experiments with SIP, and I know there's a few in the literature, but since you're presenting, can you just mention other sort of Pulsechase type experiments that have been done? Because I know a couple people looked at it going into RNA at one point, and other people looked at sort of this potential for transfer. But as far as I know, this is the most detailed one I've ever seen. Yeah,
and no, we're not the first to try it, for sure. I think that and especially now that you can do SIP with the high throughput, couple it with high throughput DNA sequencing and see things in much more detail. It's a really attractive way to look for interactions between microbes. But yeah, no, Tillman Luters has definitely done some experiments where he's looked at labeling over time. I think Tillman Luters has even done experiments where he's labeled E. coli and just put that in soil. Mm. And SIP is really starting to mature too in terms of a technique, and I think that.
I feel like the word finesse is now associated with a SIP, whereas at first it was like tossing stuff in, right?
Yeah, I think, right, there's this idea of not going for stable isotope enrichment, but going for real stable isotope probing and adding your substrates at environmentally relevant concentrations. So instead of just, hammering your community with a whole bunch of isotopically labeled substrates, you're adding things that are like somewhat relevant ecologically. And yeah, the high throughput DNA sequencing allows you to do that because it makes it so much more sensitive. Yeah.
So another question came in about position-specific DNA SIP. Would you be able to do phylotype resolved labeling at an adenine C6, for example? Or, I mean, at this point, you're just looking at total bulk DNA weight, right?
Yeah, I don't know. I mean, I wouldn't know. Yeah, right now, I mean, not the way that we do it, I don't think. I mean, I think that there's some interesting work that's being done by Bruce Hungate's group, and it's tricky, but it would be really nice. Oh well, in addition to what Hungate's trying to do with his quantitative SIP, there's a nano SIP, right, where you can really interrogate the extent of labeling by hybridizing your DNA to this sort of nano SIMS wafer, and then looking at it. That's being developed by Jennifer Petridge and applied. So maybe that would be the way to go if you wanted to get at really specific measurements of 13C incorporation.
Okay, so Matthew has a question. I can try to unmute you for asking. How do I unmute you? Darn it, I think I muted everyone by accident. Sorry.
I muted myself.
Yeah.
I was just wondering, sort of in the mindset of annotations are notoriously terrible. Have you been able to, or thought about looking at the data of the non-labeled fractions for things that should be breaking down cellulose and aren't picking it up, as in things that are either wrongly predicted or just aren't showing up?
Yeah, we haven't done that, and that sort of brings to mind this. So I think that, and at least with the xylose, I'm going to kind of digress on your question here, but with the xylose, I would imagine, if we looked at all the genomes of the microbes, or even if we cultured a lot of the microbes from our soil, that we would find many more that consume xylose than, say, just Penny-Bacillus, right? But it may be that there's other constraints on the kinds of microbes that get labeled in your microcosm than simply the genomic capacity for some function, right? So, in the way that we applied our substrate, Penny-Bacillus, because they are fast growers and they can resuscitate quickly, respond to nutrient influx really fast, that might be the actual trait that allows them to consume the xylose first. So maybe there's similar ecological attributes for the cellulose degraders beyond having a cellulase and a beta-glucosidase. So yeah, I think it would be really interesting, I think, to look at what you would expect to be a cellulose degrader versus what you saw as labeled, and then maybe try to figure out if there was some ecological characteristic that had, I'm not sure what sort of features you would look for in the genome, but if there was some ecological characteristics that differentiated those two groups. Yeah, that's an interesting question. No, we haven't done that.
Yeah, I think it's a good point, right? Because the requirement you're looking for is growth, right? And things can be using a substrate without ever needing to actually replicate their DNA.
Right. Yeah, right. So that's true too. If it's not putting the carbon into biomass, then it wouldn't become labeled in a DNASIP experiment. Or if it's not putting the carbon into DNA, then obviously we're not going to find it. Yeah.
It looks like Josh has a question.
Sure. Yeah, Chuck, I think that was a really great talk, and I love how you're interacting with your data there. I'd love to go offline and ask how you were doing that.
Yeah, sure.
But that was really neat. One thing I was curious about, so we published a paper in MBio a couple of years ago now where we used different soils, different substrates, times well. One of the differences between our experiments is you guys took, I think, what was sort of the constituents of plant biomass.
That's right, yeah.
With swapping in labeled compounds. I was just wondering if you were also tempted to just offer the labeled and not the whole suite from a plant. And sort of what was the decision-making process behind that?
Well, okay, so part of the decision-making process was to develop this method where we could interchange any number of different substrates and only use a single control. And I will say that we have done sort of this single substrate versus substrate mixture experiments. They're not published. Yeah, I think that's a really interesting point. I'm not sure what the effect would have been in our experiment. I will say that with respect to the cellulose, when we've done this from the same soils in the past, not in this same experiment, like the chloroflexi, that salvibrio type, the verruca microbia are consistently labeled, whether it's a mixture or not. But there are differences, and I'm not sure exactly what are the mechanisms behind that. Yeah, so the real decision behind the complex mixture was, if we wanted to go beyond xylose and cellulose and into the myriad of other plant biomass components, could we work with a low enough amount of label in this mixture and still see some signal?
Right.
And I think we did, but yeah, there's certainly artifacts that are apparent or that will be, yeah.
A follow-up question about your data sets. You were trying to think about why you would have a different labeled population based on your ribosomal data set, and then your metagenomic data set gave you some that you weren't expecting. And I was just curious if you have a primer bias issue, if you've checked in the metagenomic data sets to see if your primers match those particular contigs.
Right, so I will say, so I think their primer bias is a problem, and those Parkubacteria types just like what Banfield's group has been showing in their papers. You know, the primers, both based on the mismatches at the primer target and due to like insertions, these kind of like introns within the small segment ribosomal RNA genes. Yeah, so that can limit the types that we will recover based on our amplicon studies. So yeah, I don't know. I'm not sure how to get around that. I guess like if you did, I mean, I think that there's a paper out where if you try different regions, I mean, it becomes, it's difficult to expand the experiment even more. But yeah.
But in those contigs, you must have full ribosomal regions.
Yeah. So I mean, if you could do, I think. What would be interesting would be so like if you didn't want to look at your data through this prism of the PCR, it would be interesting to do metagenomics on every single fraction, and then like if you could, if you could assemble say contigs from all your data at once, and then just look at how the contigs vary in the high density fractions in terms of representation, you could treat them like we do OTUs. And that might be a way to get around working with your amplicon libraries and that primer bias. Yeah. Yeah, so I will say like the Parkii bacteria are definitely missed by our primers. And we wouldn't know if they were labeled. Yeah.
So because you've cited Noah and Paul's work a couple times already, so going back to that, right, with this idea of relic DNA in your soils,
yeah.
Is there any thought, right, with this initial burst that you're seeing in the organisms, right? Would they die off and leave DNA, or is any of the mismatch maybe related to this idea that there's chances for DNA having a longer lifetime? And at that point, you'd probably see it in a ribosomal pool and less in a metagenome.
Yeah, so I mean, I think okay, so I'll say a couple things. I think that, there are certainly benefits to doing the ribosomal RNA versus the DNA, and that's like, stemming from this fact that the DNA might like carry that label for a really long time. But one of the nice things about CIP though is we can kind of cut through that background because we're looking for activity. So I wouldn't.
Yeah, but in 7 days you probably have an organism.
Yeah, I will say
die, right? And
yeah, but so the Pepe-Bacillus was labeled. So there were like. So for the most part, say the Pepe-Bacillus OTUs, they were labeled at day 1, and then they didn't appear labeled at day 3 and 7 and 14 and 30. But there were OTUs, like a limited number of exceptions to that. So there was at least one Pepe-Bacillus OTU that like continued to be labeled beyond day 1. And who knows what's going on there? That could represent just like DNA that's being preserved and in some way, who knows, relic DNA maybe. But like for the most part, we saw the labeling kind of go up and then go down across time.
Oh, which is interesting, right? Because your label's still in the system.
Yeah, yeah. Well, some of it is.
Where did it go? Like your mass, like, were you losing mass? Or?
Well, we definitely, so like.
Sorry, we should let you go. Like, I'm sure you have other things to do today, but.
We definitely, I mean, a lot of it's respired, right?
Yeah.
So we lost like 70% of our 13C to CO2 within 24 hours.
Oh, wow.
Yeah, so it doesn't actually, it's not like recycled back into the community, at least in the way that we set it up. Sometimes that can be a problem if you're probing for autotrophs, say, with like labeled CO2. But in our case, it wasn't a problem.
Cool. All right. Well, does anyone else in the Hangout have another question? Okay. Anyone on YouTube? I haven't seen anything come in. Of course, Chuck's available on Twitter. We didn't see lots of relic DNA in the exos we used. Okay. Well, Paul has told us that agrees with what we've seen. So I'm going to stop the YouTube feed, and so anyone who has questions can ask Chuck. He's Chuck_PR on Twitter. And thanks for doing this, Chuck.
Yeah, it was really great. Thanks for having me.